I'll give you a little overview of the kinds of things we're trying to do with the CCBSC program in the ND ACES larger project. What we're interested in doing is figuring out whether cells behave differently when they're grown in 3-dimension versus 2-dimension. And our particular interest in wanting to grow these cells in 3-dimension is because a number of studies have shown that cancer cells behave very differently when they're grown in 3-dimension, so when they're in the form of sort of a tumor, than they do when they're flat in 2-dimension. And that's typically the way we grow them in cell culture in the laboratory, is just flat on a cell culture dish. And so it turns out that the way cells behave in response to drug treatments are not the same in those two formats. So if we really want to study something that's more relevant to the human condition and tumors in a person's body, we really want to try to grow them in 3-dimension. So that's one of the things that we're trying to do with this CCBSC program, is to design the types of materials that will support cell growth in 3-dimension, and then observe how the cells behave when they're growing in 3-dimension, and then hopefully design a way that we can not just model the way tumors grow better, but also develop ways to target those tumors, those, again, 3D cultures, not really tumors, but as tumor-like as we can make them, and find ways that we can try to stop their growth or kill them, and things that will be ultimately useful for patients. And we have a combination of computational scientists, and then we have material scientists, and then we have cell biologists all working together to try to design the best materials, build the best materials, and then grow cells in those materials in a way that's going to accurately model the way a tumor behaves in an individual's body. And we're focusing currently on prostate cancer and breast cancer. Certainly there are other forms of cancer, but our immediate interest is in those two forms of cancer. And we're interested in how what we call the primary tumor develops in the prostate or the breast, and then how it metastasizes to bone. So we're looking at 3-dimensional cultures of both soft materials that would model the prostate or the breast, and then hard materials that would model bone. And in both of those cases, we want to know how are the cells behaving in those environments, and how can we specifically sort of attack those cells in those environments. And that's a lot of the projects will revolve around those sorts of interests.